Hello guys and welcome back to Excel Academy. So today we will be doing regulation of glycolysis. So till now we talked about glycolysis, like different substrates of glycolysis, gluconeogenesis, right? And now we are going to discuss the regulation of glycolysis. So all of the reactions of glycolysis and most of the reactions of gluconeogenesis, it occurs in the cytosol. So yes, we know that glycolysis and gluconeogenesis occurs in the cytoplasm. Nearly all of the reactions of glycolysis and gluconeogenesis are reciprocally regulated, yes, only except the three steps, right? They all are going to be same. The result of this reciprocal regulation allows the cell to rapidly catabolize glucose when the cellular energy supply is low and to synthesize and store glucose when the energy level of the cell is high. है ना? इसलिए it is easy for the cell to undergo glycolysis and gluconeogenesis because they all are having the same number of enzymes and same number of enzymes and the weight. Okay, except only those three reactions. Rest all enzymes and rest all steps are going to be similar. Now, regulation is in two ways. The first is the kinetic property of its hexokinase isoenzymes. Second is the allostatic regulation of the enzymes that catalyze the three irreversible reactions. So these are the only three irreversible reactions which are different in both glycolysis and gluconeogenesis. First one is from your hexokinase, phosphofructokinase, and the pyruvate kinase. So all three of these enzymes are allosterically regulated. So what do you mean by allosterically regulated, Pallavi? Yes. What does it mean when an enzyme is allosterically regulated? What does it mean? It means that there is another binding site on which the inhibitor or any other activator can bind and regulate the action of the enzyme. Okay. So gluconeogenesis, it replaces these irreversible reactions with the corresponding irreversible reaction that are exergonic in the direction of glucose synthesis. So again, you have only different exergonic reactions. Exergonic reaction is it is energy releasing.
Energy release.
Yes, exergonic reaction means it is energy releasing. Okay, so the three bypass reaction of gluconeogenesis is catalyzed by glucose 6-phosphatase, fructose 1,6 bisphosphatase, and the last one है आपका pyruvate carboxylase and pep carboxykinase, right? Now, glucose 6-phosphatase is not under allostatic control. This enzyme is located in the lumen of endoplasmic reticulum, right? ये भी यहाँ से important हो गया, ठीक है? क्या क्या याद रखना है हमें? So the Km of glucose 6-phosphatase for glucose 6-phosphate is higher than the cellular concentration of glucose 6-phosphate. It means that it has very low affinity towards its substrate, है ना? हमने यही पढ़ा ना, when Km is high, it means its affinity is low. So the Km of glucose 6-phosphatase for glucose 6-phosphate is higher, so it means it has got less affinity. As a result, glucose 6-phosphatase show a linear dependence of activity as a function of glucose 6-phosphate concentration. This enzyme is said to be under substrate level control. What does it mean? What do you mean by this statement? That glucose 6-phosphatase show a linear dependence of activity as a function of glucose 6-phosphate concentration, and it is said to be under substrate level control. What does it mean?
Ma'am, that means direct involvement of ATP instead of NADPH and FADPH.
Okay, good. Yes, very good. Okay, now, so this is how the regulation is happening. So let's see like what are the positive regulators and the negative regulators. Okay, so here we have got the glucose, and then here hexokinase, phosphofructokinase, and pyruvate kinase, and these three In place of in the case of gluconeogenesis, we have got pep carboxykinase, pep pyruvate carboxylase, fructose 1,6 bisphosphatase and glucose 6 phosphatase. ठीक है, अभी हमें ये देखना है कौन से ऐसे molecules हैं which are going to act as the activators and which are going to act as the inhibitors. ठीक है, so of course for the activator of hexokinase, glucose 6 phosphate. Glucose 6 phosphate has got negative impact. Why? Because if you have got high concentration of glucose 6 phosphate, then the cell understands that there is already enough amount of ATP is there, right? So it will not it will show like so glucose 6 phosphate concentration is going to have the negative impact on the hexokinase. Agree? Pallavi, do you agree?
Yes, ma'am.
Okay. So similarly, phosphofructokinase, ATP citrate has got negative, whereas fructose 2 bisphosphate and AMP has got the positive effect, right? So AMP, if it is high, what does the cell will understand that it does not have the ATP. So of course it is going for the glycolysis. Okay, so we need to have the AP sorry. So we need to have the AMP concentration high. Okay. Then again, pyruvate kinase. So of course, ATP, acetyl-CoA, alanine, they all are going to have the negative impact, and fructose 2 bisphosphate and AMP is going to act as the positive regulator of the pyruvate kinase. So is it clear to you why glucose 6 phosphate, AMP, ATP are positive and negative respectively? Is this part clear to you, Pallavi?
Yes, ma'am.
Okay. Now coming to the gluconeogenesis. So here also, again, so we have got the pyruvate carboxylase. Acetyl-CoA is having more concentration. So what does it mean? Acetyl-CoA अगर आपका ज़्यादा है, so cell understands that it already have enough concentration of ATP. क्योंकि acetyl-CoA मतलब वो आपका Krebs cycle में जाता है, ETC में जाता है, you have got high amount of energy, right? So if it is more, then it is going to give a positive feedback onto the pyruvate carboxylase. So what does it mean? It has got more acetyl-CoA. So अभी हमें और glucose को glycolysis करने की जरूरत नहीं है. So I have got enough energy. So what I can do is now I can convert excess of now I can convert excess of the acetyl-CoA back to the glucose synthesis. है ना? So that's why it has got the positive effect on the pyruvate carboxylase. Then of course, AMP, fructose 2 phosphate अगर ज़्यादा है, so then again what will happen? It is having the negative impact on the bisphosphatase. यानी कि और मत glucose synthesize करो. AMP अगर ज़्यादा है, so it means glucose synthesize मत करो, right? So this is how these are the molecules which are acting as the activators and the inhibitors in the regulation of the glycolysis. Clear है Pallavi?
Yes, ma'am.
सिंपल है, बस अगर ये फ्लो चार्ट याद रहेगा तो बहुत इजी हो जाएगा. जो भी आगे क्वेश्चंस आते हैं ना, वो आपके भी इजी हो जाएगा. ओके, अब लेट्स टॉक अबाउट दीज एंजाइम्स इन डिटेल. अभी तक हमने जो भी फिगर में देखा है उसको थोड़ा सा उसको हम डिटेल में और पढ़ रहे हैं. So the hexokinases. So there are 4 types of hexokinases which are present in the animals. Hexokinase 4. It is also known as glucokinase. So it means it is going to be specific only for your glucose. It is found in the liver. Now hexokinases 1, 2 and 3 are present in different tissue and bind reversibly to an ion channel called a porin in the outer membrane of the mitochondria. Okay, so along with that, hexokinase 1, 2, 3, we have got the porin. Now, these isozymes have high affinities for glucose related to its concentration in the blood. Their K value is 0.1 millimolar, which is less than the concentration of glucose in the blood. So, उसका K value आपको दिया गया है. So, this indicates that hexokinase has high affinity for glucose. क्यों? Because उसका Km value आपका कम है. So affinity उसका high हो गया. Okay. So hexokinase 1, 2, 3 are blocked by glucose phosphate. When in addition, Hexokinase 1, 2, 3 are inhibited by phosphorylating glucose molecules by glucose 6-phosphate. पल्लवी ये सेंटेंस क्लियर हो रहा है कि हेक्सोकाइनेज 1 2 3 आर ब्लॉक बाय ग्लूकोज 6-phosphate. ये सेंटेंस क्लियर है क्यों उसको वो ब्लॉक कर रहा है, इनहिबिट कर रहा है? When the blood glucose level is high, it will not phosphorylate all glucose molecules.
Yes, ma'am.
Okay, हाँ, same thing. कि अभी जब ज्यादा जरूरत नहीं है, there is no need to undergo the process. Okay. now glucokinase. तो अभी हमने क्या देखा था? Hexokinase has got the affinity of around 0.01 millimolar. ठीक है? Now glucokinase has km of about 10 millimolar. So it is more than circulating concentration of glucose in the blood. It is not inhibited by glucose 6-phosphate. This will allow the conversion of excess glucose into glucose 6-phosphate by the liver. Later, what happens is that glucose 6-phosphate is directed to the glycogen synthesis. So it is believed to be a glucose sensor because glucokinase does not need to work at maximum velocity. It is highly sensitive to small changes in the blood glucose. Since उसका Km इतना हाई है तो ग्लूकोकाइनेज में एफिनिटी कम रहेगा, ठीक है? तो कितना भी आपका ज्यादा ग्लूकोज आपका ब्लड लेवल में बढ़ जाए, ठीक है? तो सपोज आपका ब्लड लेवल जो है उसका ग्लूकोज इज इंक्रीजिंग. So because of this increase in the glucose concentration, the glucokinase GK is not going to get affected. Okay, inhibit नहीं होगा. उल्टा वो क्या करेगा? The excess glucose will be converted into the glycogen synthesis. अभी क्लियर है? Hexokinase के केस में क्या है? If you have got the high concentration of glucose,
Km value is
हाँ, it is less. So what will happen? अगर आपके पास glucose concentration high होगा, तो hexokinase जो है वो आपका inhibit हो जाएगा, ठीक है? Whereas in the case of glucokinase, it will not get inhibited. उल्टा वो excess glucose को glycogen storage के लिए redirect कर देगा, ठीक है? Clear है? How glucokinase and hexokinases are
yes, ma'am.
Okay, good. Important है ये. Now, during hyperglycemic condition, so hyperglycemic condition means you have got the more concentration of glucose, right? In the blood, hyperglycemic होता है आपका high concentration of glucose in the blood, right? So insulin is released which trigger the synthesis of glucokinase. अब insulin क्या करेगा? Glucokinase का synthesis increase करेगा. Glucokinase will go and bind to the glucokinase regulator protein. Now this entire complex, glucokinase regulator protein and आपका glucokinase, ठीक है? ये सारा क्या करेगा? आपका सीधा न्यूक्लियस में जाएगा, ठीक है? कहाँ पे? न्यूक्लियस ऑफ द लिवर, and the phosphorylation of glucose to glucose 6-phosphates are prevented as this reaction occurs in the cytoplasm. Now this complex formation is a trigger in a high concentration of fructose 6-phosphate. Now when the blood glucose level आपका increase होगा, GKRP यानी कि glucokinase regulator protein will release glucokinase, and glucokinase moves back to the nuclear pose and again phosphorylate glucose. Clear?
Yes, ma'am.
है ना? सिंपल है कि किस तरह से आपको रेगुलेशन हो रहा है. तो यहाँ पे like to understand about glucokinase का activity, we have got another protein called glucokinase regulator protein. ठीक है? तो ये क्या करेगा? ये पूरा कॉम्प्लेक्स जो है न्यूक्लियस ऑफ द लिवर में जाके बैठ जाएगा, ठीक है? एंड द फॉस्फोराइलेशन ऑफ ग्लूकोज ग्लूकोज 6-phosphate नहीं होगा क्योंकि वो आपका कहाँ होता है?
साइटोप्लाज्म में होता है.
हाँ, एंड अभी क्या है? आपका ग्लूकोकाइनेज आपका न्यूक्लियस में है. जैसे ही आपका कॉन्सेंट्रेशन बढ़ता है वैसे ही आपका ग्लूकोकाइनेज जो है वापस आ जाएगा and then it will do the phosphorylation of glucose. Clear?
Yes, ma'am.
Now regulation of PFK1. अभी हमने क्या देखा था? Regulation of hexokinases. अभी now the second enzyme that is phosphofructokinase. Now it has a homotetrameric PFK1 has a homotetrameric enzyme which is composed of 4 identical sequences. तो अभी ये कैसे दिखता होगा? So let us imagine that it is something like this. Not like this. Okay, so we have got four identical subunits like any other allosteric protein. जैसे hemoglobin के आपका क्या है? Homotetrameric protein, right? And the enzymes, the binding of allosteric effectors and substrates communicated with each of the active sites. अब इसका क्या मतलब होता है ये सेंटेंस का? It means that the moment एक subunit को आपका substrate bind करता है, it will help in the binding of the substrate to the other units. ठीक है? तो क्या होगा? The binding of the substrate to the other subunit becomes faster. इसको हम क्या बोलते हैं? Cooperative binding. Cooperative binding. So PFK exists in two sets of conformation called T and R. यहाँ पे T मतलब क्या हुआ आपका? Tense form. And R क्या होगा फिर? Relaxed form. Okay. Now these two conformational states of the PFK1 are in equilibrium with each other. So after tense form and relaxed forms are in equilibrium. Okay. So PFK1 is allostatically inhibited by high levels of ATP in substrate. ये हमने अभी देखा था, right? Inhibited क्यों? Because अगर आपके पास ATP in substrate concentration ज़्यादा है, तो आपको glycolysis करने की जरूरत नहीं है, and that's why it is going to inhibit the activity of PFK1. Okay. Now, AMP is an allosteric activator of PFK1. It reverses the inhibition due to high concentration of AMP. So AMP binds preferentially to the R state of the PFK. वो क्या करेगा? R state relaxed form में आपका AMP को bind करेगा. Okay, so what will happen now? आपका जो relaxed form है, तो ये सारे आपके R form में हैं. Let us imagine that ये सब आपके R form में हैं. The moment ATP starts binding, what will happen? This R will start getting converted to T form. Clear हो रहा है पल्लवी? से कैसा होता है आपका जो प्रोटीन है रिलैक्स फॉर्म में, यानी कि no substrate is bound. The moment ATP comes in binds, आपका जो R है वो आपका tense form में चला जाएगा. Tense form means simply the proteins are bound with their substrate. ठीक है? Okay, so what will happen? Then the PFK1 is going to do its function. क्यों? क्योंकि अभी मालूम था उसके पास AMP present है और AMP का हमें क्या करना है? We need ATP, so it will be active. So it will be inhibited. ठीक है? So it is an activator of the PFK1. So you have a two molecules of ADP and ATP plus AMP. ठीक है? So the equilibrium constant is given by ATP AMP divided by square of ADP. ये इक्वेशन में हम ऐसे लिख सकते हैं अगर मुझे इसका इक्विलिब्रियम कांस्टेंट लिखना हो. ठीक है? ओके. Now AMP in the cell is formed by the enzyme adenylate kinase. ये हमें मालूम है adenylate kinase, right? So a small decrease in the concentration of ATP can drastically increase the level of AMP. Yes, अगर आपका ATP decrease हो रहा है तो आपका AMP increase होगा, obviously बात है. So the steady state concentration of ATP in the cell is 10 times greater than the concentration of ADP, 50 times concentration of AMP. As a result of the activity of adenylate kinase, 10% decrease in the concentration of ATP results in 400% increase in the concentration of AMP. अंतर दिख रहा है? क्यों ऐसा?
Yes, ma'am.
अभी हमने वो फॉर्मूला अभी भी देखा है, राइट? और अगर 50% डिक्रीज हो रहा है, सॉरी 10%, 10 टाइम्स ग्रेटर देन द कंसंट्रेशन ऑफ योर ADP, राइट? और यहाँ पे क्या 50 टाइम्स द कंसंट्रेशन ऑफ AMP. अगर 10% डिक्रीज हो रहा है, so what will happen? There will be a 400% increase in the concentration of AMP. So अभी हमें क्या मालूम है? That AMP, ADP ये सब आपके effector है of the PFK1. So PFK1 is dependent on the ATP, ADP, AMP concentrations, which are all the functions of the cellular energy status. See, ये क्यों energy status बताते हैं? ATP concentration high रहेगा, so it means your cell is having good amount of nutrition, so it is going to grow and divide. अगर आपका एटीपी चलता है, बिकॉज हमें पता है कोई भी सेलुलर एक्टिविटी के लिए एटीपी लगता ही है, ठीक है। सिमिलरली अभी मेरे पास एडीपी ज्यादा है और एएमपी ज्यादा है, ठीक है। सो नाउ द सेल इज नॉट गोइंग टू डू और एनी कॉस्ट इन्वॉल्विंग प्रोसेस। अभी वो क्या करेगा कि एनर्जी कम है, सो वो अपनी एनर्जी स्टोर करने में या किसी तरह से इसको बनाने में लग जाएगा, ओके। तो ये आपका एनर्जी सेलुलर स्टेटस बताता है। So let's see the figure over here. So we have got the fructose-6-phosphate millimolar and phosphofructokinase activity. Okay. Since we are talking about PFK1, that's why we have got the fructose-6-phosphate. Now look at the concentration over here. This is concentration है 1 millimolar ATP, right? 1 millimolar ATP and 0.1 millimolar AMP. यहाँ पे कुछ भी inhibitors नहीं है, okay? And very low concentration of ATP. Can you see the difference in the activity of PFK1, phosphofructokinase? So which one is showing the maximum activity?
Ma'am, with no inhibitor, low ATP.
Yes, it is showing the maximum activity at very low concentration of your fructose-6-phosphate. Yeah? So from this you can understand like how exactly it is dependent. अगर उसको लगता है बिल्कुल भी ATP नहीं है, then the PFK1 activity is going to be high. अगर उसको मालूम है कि activity थोड़ा सा ATP available है, so it is going to have the less activity. क्योंकि वैसे भी ATP जो है वो आपका नेगेटिव इफेक्ट करता है, राइट? इट इज अ नेगेटिव रेगुलेटर ऑफ योर PFK1, है ना? यहाँ पे क्या थोड़ा सा AMP डाला था, that's why थोड़ा सा increase in activity था. When you had no ATP, then you can see that it is having the maximum activity at very less concentration of your fructose-6-phosphate. Okay, so Pallavi, this figure is clear to you?
Yes, ma'am.
Okay. Now ATP feedback inhibition. So the function of the glycolytic pathway is to generate the ATP. Yes. So ATP is going to act as both substrate as well as the allosteric inactivator. ये भी हम agree करते हैं, right? क्योंकि कहीं-कहीं पे वो allosteric bind करके activate inactivate करता है. And the enzyme has got two binding sites for ATP. One is the substrate binding site, the other one is the inhibitory site. अभी क्यों? Because जो भी हमने स्टेप की बात की वहाँ पे आपका एनर्जी यूटिलाइज होता है, ठीक है। तो यूटिलाइज होने के लिए एक जगह वो सबस्ट्रेट का काम करेगा और एलिस्टिकली वो इसको एक्टिवेट या इनहिबिट करेगा. सो द PFK1 इट हैज गॉट द टू साइड्स, P and R स्टेट है. सो द इनहिबिटरी ATP बाइंडिंग साइट ओनली बाइंड्स ATP व्हेन द एंजाइम इज इन T कॉन्फॉर्मेशन. ठीक है, अभी हमने क्या देखा था, जब आपका R स्टेट होता है तब कौन बाइंड करता है? AMP. ठीक है, T स्टेट है तो कौन बाइंड कर रहा है आपका? ATP.
Substrate. ATP.
हाँ. So the other substrate fructose-6-phosphate binds only to the R. Okay. So the high concentration of ATP should be equivalent towards the T conformation, which decreases the affinity of the enzyme for fructose-6-phosphate. क्यों? क्योंकि अभी मुझे और fructose-6-phosphate की जरूरत नहीं है, क्योंकि ऑलरेडी मेरे सिस्टम में ATP बहुत ज्यादा है. ठीक है. Now fructose-1,6-bisphosphatase, it is another important site for gluconeogenesis regulation. High concentration of citrate, okay, high concentration of citrate activates fructose-1,6-bisphosphatase and high concentration of AMP inhibit it. क्यों? अभी हम gluconeogenesis की बात कर रहे हैं, right? तो gluconeogenesis मतलब I am going to synthesize glucose. तो अगर मेरे पास citrate ज्यादा है, then only fructose-1,6-bisphosphatase काम करेगा, because मुझे ग्लूकोज बनाना है, right? तो मुझे citrate ज्यादा होगा तो ही मैं ग्लूकोज बना पाऊंगी. Are you getting it?
Yes, ma'am.
And high concentration of AMP inhibit. Okay. So AMP enhances the inhibition of fructose-2,6-bisphosphate. Allosteric effects of fructose-1,6-bisphosphatase and phosphofructokinase are common. So these effects reciprocally regulate the enzyme. So ये भी हमने देखा ही था in the figure which we saw. So there we have talked about the fructose-2,6-bisphosphate and AMP, right? So they are reciprocally regulating the whole enzyme. So okay, so fructose-6-phosphate, fructose-1,6-bisphosphate. तो हमने यहाँ पे भी क्या देखा? AMP हो गया, fructose-2,6-bisphosphate and citrate. So both of these two are having the positive effect and यहाँ पे citrate आपका negative दे रहा है. And over here उल्टा, citrate आपको positive रहेगा, बाकी जो आपका negative effect देगा. ठीक है? So this provides a way for gluconeogenesis and glycolysis to be coordinated such that when one pathway is active, the other has to be inactive. Obviously, right? एक जगह अगर आपको ग्लाइकोलाइसिस हो रहा है तो आपको ग्लूकोनियोजेनेसिस बंद होना चाहिए. दोनों एक्टिवेट रहेंगे तो नेट रिजल्ट क्या होगा? दैट विल बी लॉस होगा. So if both pathways were active at the same time, the net result would be the net hydrolysis of 2 ATPs and 2 GTPs per reaction cycle. तो हमने देखा ही था कि अगर मैं नेट दोनों ग्लाइकोलाइसिस और ग्लूकोनियोजेनेसिस करूंगी, so at the end I am using extra 2 ATPs and 2 GTPs per reaction cycle. So I am in the loss only. ठीक है? So since both pathways are highly exergonic, so there is no thermodynamic barrier to such cycling, as it is shown. Now this one, okay, the cycling of fructose 6-phosphate and fructose 1,6-bisphosphate, it is called the substrate cycling, and the net result of the substrate cycling is the hydrolysis of ATP. क्या मेरा ATP use होता है, ठीक है? So finally the thing is that सेल हमारा इस तरह से काम करता है ताकि वो हमारे ATP को वेस्ट ना करे. Okay, so they are very regulated, very economical. It's not like the way we spend money. हम कभी सोचते नहीं है कि it's okay और आ जाएगा, ठीक है? But cells are not like that. We thinking that now let me use excess amount of energy and later I can synthesize or I can generate more. ऐसा नहीं है. जब भी जरूरत होती है वो टाइम यूज करता है. Okay, Palak, are you getting this?
Yes, ma'am.
Okay. Now, fructose 2,6-bisphosphate, it is synthesized by phosphofructokinase 2. अब यहाँ पे थोड़ा अंदर देखना, ठीक है? अभी तक हम क्या डिस्कस कर रहे थे? फ्रक्टोज 1,6-bisphosphate and about the PFK1. अभी ये क्या है? फ्रक्टोज 2,6-bisphosphate है and it is phosphofructokinase 2. ठीक है? थोड़ा सा different enzyme है ये. So during hyperglycemic condition, again you have got the glucose concentration high. The insulin hormone increases the activity of PFK2. अब PFK2 produces fructose 2,6-bisphosphate, which work as an allogenic activator of PFK1 and activates glycolysis. है ना? क्योंकि देखो आपके पास ग्लूकोज कंडीशन ज्यादा है, तो व्हाट विल हैपन? इंसुलिन आपको बढ़ जाएगा. इंसुलिन क्या कर रहा है? PFK2 की एक्टिविटी बढ़ा रहा है. PFK2 की एक्टिविटी बढ़ने से क्या होगा? PFK1 भी आपका एक्टिवेट हो जाएगा और ग्लाइकोलाइसिस होगा. Whereas fructose 2,6-bisphosphate also decreases the inhibitory effects of the ATP. अब ये क्या देखोगे? You have got the more glucose in the blood level and more hormone will be there. ठीक है? तो जैसे जैसे आपका concentration यहाँ पे देखो, यहाँ पे क्या वैल्यू है? 0.01 माइक्रोग्राम. जब आपने थोड़ा सा concentration बढ़ाया तो आपकी एक्टिविटी भी बढ़ गई. Then when you have the fructose 2,6-bisphosphate, तो आपकी एक्टिविटी और ज्यादा बढ़ती है. So here we are talking about the activity of your PFK. This is another enzyme which is responsible for the glycolysis regulation. इतना important नहीं है, but it is important to know about fructose 2,6-bisphosphate. Now, PFK2, it is a bifunctional enzyme which behaves both as phosphatase and kinase. It can remove phosphate group, it can add phosphate group, right? So insulin hormones activate the kinase activity of your PFK2 and glucagon जो है वो आपका phosphatase activity of PFK2 करेगा. So if you remember in the previous lecture we have talked about that इंसुलिन जो है आपका पॉजिटिव इफेक्ट करता है ग्लाइकोलाइसिस है ना? तो
Yes, ma'am. Yes, ma'am.
इंक्रीज इन द इंसुलिन लेवल इट मींस ग्लाइकोलाइसिस होगा. और अगर हमारे पास ग्लूकागॉन है तो आपका ग्लूकोनियोजेनेसिस होगा. करेक्ट? सो एक और बात. सो इंसुलिन हार्मोन एक्टिवेट्स काइनेज एक्टिविटी ऑफ योर PFK2, जिसकी वजह से क्या होगा? आपका PFK1 एक्टिवेट होगा एंड ग्लाइकोलाइसिस होगा. Whereas glucagon activates phosphatase activity, यानी कि ग्लूकोनियोजेनेसिस की तरफ जाएगा. ठीक है? So when phosphorylated in response to the hormone glucagon, it is released into the blood in response to the low blood sugar. उसके पास कम है, right? It function as kinase when dephosphorylated in response to the insulin. Fructose 2,6-bisphosphate, it is produced via Hormone induced covalent modification of PFK2 as an indicator of high levels of available glucose and allosterically activates PFK1. Accumulated fructose 1,6 bisphosphate activates pyruvate kinase, providing feedback mechanism, feedback control आपको देखना है। अभी यहाँ पे देखिए कैसे होता है। So you have got the fructose 6 phosphate, PFK2 active हुआ, आपको मिला fructose 2,6 bisphosphate. Phosphatase will give you again back to fructose 6 phosphate and आपका extra जो phosphate है वो remove हो जाएगा। अब यहाँ पे effect of hormones है, यहाँ पे क्या है आपका हार्मोन लेवल का रेगुलेशन है। So what do we see? इंसुलिन जब रहेगा so it is going to be positive. So आपका PFK2 है जो है क्या होगा, इनएक्टिव हो जाएगा। कौन सा आपका, ये आपका इनएक्टिव हो जाएगा if you have got the insulin over here, ठीक है। Now ग्लूकागॉन जो है ATP यूज करके यहाँ पे उसको इंड्यूस करेगा, कौन सा आपका काइनेज एक्टिविटी। यहाँ पे क्या है आपका फॉस्फेटेज एक्टिविटी। कैन यू सी दैट, कैन यू अंडरस्टैंड नाउ? इंसुलिन जो है फॉस्फेटेज एक्टिविटी को इनहिबिट करता है, यानी कि उसको इनएक्टिव कर देगा, ठीक है। ग्लूकागॉन अगर है तो वो उसकी काइनेज एक्टिविटी को इनहिबिट करता है, ठीक है। उल्टा, यानी कि इंसुलिन है तो मुझे PFK2 एक्टिव चाहिए, यानी कि इसका फॉस्फेटेज जो पार्ट है उसको इनएक्टिव कर देगा। ग्लूकागॉन है तो उसके काइनेज एक्टिविटी को इनहिबिट करता है क्योंकि उसको फॉस्फेटेज पार्ट चाहिए, ठीक है। क्लियर है या कंफ्यूजन आई है पल्लवी?
No, ma'am, it's clear.
It's clear ना? So just understand about the kinase and phosphatase activity. Okay. Yeah. Now, so PFK2 in turn activated by the dephosphorylation reaction catalyzed by phosphoprotein phosphorylase triple P की जो भी हमने देखा था। So this is the action of the insulin. So insulin जो है वो आपके phosphoprotein phosphatase पे काम करेगा and that's how it is going to activate your PFK2, ठीक है, to be phosphatase। अब क्या होता है, very simple, जो एंजाइम आपका काइनेज एंड फॉस्फेटेज ग्रुप एक्ट करता है उसको भी एक्टिवेट करने के लिए या डीएक्टिवेट करने के लिए यू कैन ऐड फॉस्फेट ग्रुप, ठीक है। तो ये फॉस्फेट ग्रुप कौन ऐड कर रहा है, आपका फॉस्फोप्रोटीन फॉस्फेटेज। आपको फॉस्फेटेज रिमूव करेगा एंड देन वी हैव गॉट द डिफरेंट काइनेज एक्टिविटी व्हिच कैन ऐड द फॉस्फेट ग्रुप। So insulin hormone activates phosphoprotein phosphatase enzyme which dephosphorylates PFK2 and the PFK2 का phosphate हमने हटा दिया, it becomes active and it is act as kinase, ठीक है। अगर मेरे पास PKA है, अभी जब हमने प्रीवियस में देखा था, प्रीवियस स्लाइड में हमने क्या देखा था, ये आपको दी PKA। PKA क्या है आपका प्रोटीन काइनेज A, तो ये आपका काइनेज एक्टिविटी को सॉरी ये क्या करते हैं आपके PFK2 को काइनेज फॉस्फेट ग्रुप ऐड करता है, ठीक है। सो इट इज गोइंग टू मेक इट इनएक्टिव एंड एक्टिव, सो डिपेंडिंग ऑन द ग्लूकागॉन या इंसुलिन जो आता है। सो ग्लूकागॉन, सपोज ग्लूकागॉन है, इट इज रिलीज बाय द पैनक्रियाटिक अल्फा सेल्स व्हेन ब्लड ग्लूकोज लेवल इज लो। बीटा सेल्स आपके क्या प्रोड्यूस करते हैं, इंसुलिन। पैनक्रियाटिक बीटा सेल्स प्रोड्यूसेस इंसुलिन, पैनक्रियाटिक अल्फा सेल्स प्रोड्यूसेस ग्लूकागॉन, ठीक है। Now it is going to activate the phosphatase function of PFK2, thereby reducing the level of fructose bisphosphate in the cell. Okay, so glucagon works through GPCR pathways and its effects are mediated by cyclic AMP. Cyclic AMP क्या करेंगे? It is going to activate PKA. PKA is going to have the initiate a signal cascade of phosphorylation dephosphorylation reaction. PKA phosphorylates PFK2. And this will work as phosphatase and convert fructose 2 bisphosphate into fructose 6 phosphate, ठीक है। सिंपल, ग्लूकागॉन अगर
yes,
है तो
yes.
क्या होगा आपका PFK2 जो है फॉस्फेटेज जैसा काम करेगा, ठीक है, एंड कन्वर्ट फ्रक्टोज 2 बिस्फॉस्फेट टू फ्रक्टोज 6 फॉस्फेट। सिंपल, इंसुलिन का जो काम है उसका उल्टा काम ग्लूकागॉन करेगा। मसल एंड एडिपोज टिश्यू हैव इंसुलिन सेंसिटिव ग्लूकोज ट्रांसपोर्टर्स। Insulin promotes translocation of glucose. Insulin works through RTK pathways. So RTK, what is RTK pathways? ये नहीं मैंने अभी तक पढ़ाया है। So you know about GPCR, right? So when we were doing about the cell signaling, हमने GPCR pathways के बारे में पढ़ा है, G protein coupled receptors. Now, when you talk about RTK it stands for receptor tyrosine kinase. ठीक है? So the receptors जो रहेंगे इसके जो receptors रहेंगे RTK, तो insulin works through RTK, यानी कि क्या आपको जो प्लाज्मा मेम्ब्रेन रहेगा, ठीक है, यहाँ पे प्लाज्मा मेम्ब्रेन रहेगा, यहाँ पे जो receptors रहेंगे इसके टायरोसिन ग्रुप में काइनेज एक्टिविटी रहेगी. It means what? To the tyrosine they can add phosphate group and gets activated. ठीक है, सिंपल में बता देंगे, सिंपल भाषा. So you have got the plasma membrane here. यहाँ पे आपकी receptors, ठीक है, and through the receptors you have got the kinase domain. Kinase domain क्या करेंगे? Through the tyrosine group over here, they are going to add the phosphate. So when they add phosphate group, what happens? They get activated. इस तरह से काम करते हैं, ठीक है? Okay. So insulin works through RTK pathway, and the signal cascade activates transcription factor SREBP1, which is a sterol regulatory element binding protein. So this will increase the synthesis of glucokinase and pyruvate kinase. Citrate binds preferentially to the TS2PFK1, and hence high concentration of citrate is required. So now if you see how beautifully all these components like citrate हो गया आपका, ATP हो गया, AMP हो गया, fructose 2 बिस्फॉस्फेट हो गया, कैसे ये आपके एक्टिविटीज को on and off कर रहे हैं, ठीक है? So that is all overall just regulating the activity of your enzymes. So AMPK, a metabolic master switch, okay? So this is the allosteric regulation of glycolysis. अभी तक जो भी हमने डिस्कस किया, so हमने क्या क्या डिस्कस किया? Hexokinase, PFK1 and pyruvate kinase. So AMP activated protein kinase, AMPK. It is an enzyme which plays central role. It is activated when the cell has got high AMP to ATP ratio. See, अगर आपका AMP to ATP ratio high है, what does it mean? Numerator high है, यानी कि AMP concentration high है, ठीक है? So it is involved in AMPK is involved in lipid glucose metabolism. Once activated, it is going to phosphorylate the proteins and enzymes. Now, this switches off the anabolic pathways. AMPK switches off the anabolic pathways, यानी कि protein and lipid synthesis को वो switch off करेगा, and switches on the catabolic pathways. क्यों? AMP आपका high, AMP
because AMP is high.
Yes, very good. So AMP high है, इसलिए इसका मतलब we need more of ATP, and that's why it is going to switch on the catabolic pathways like glycolysis and fatty acid oxidation, जिससे हमें ATP मिलेगा. Good. Now AMPK promotes glycolysis in cardiac and skeletal muscle during exercises. Okay. It increases the number of glucose transporters to the plasma membrane, and hence we need more glycolysis. यानी कि it is going to stimulate more glycolysis by activating PFK. Yeah, so we have got the enzyme, hexokinase, PFK1, pyruvate kinase, activator and inhibitor का table भी दिया हुआ है. Okay. So Pallavi, is this much clear till now?
Yes,
What we have discussed for the regulation of glycolysis?
Yes, ma'am.
Okay. So I'll be stopping here now because I have prepared this much for today's class. Okay.
Yes,
So any other doubt in between you came across?
no, ma'am.